Hi. Now we will discuss about, in terms of engineering aspects, how to manufacture monoclonal antibodies and differences between monoclonal and polyclonal antibodies. So, how to manufacture monoclonal antibody for clinical use? So we can use antibody for clinical therapy. So for that, we want to first understand antibodies are produced by the B cell or B lymphocytes inside our body. So for example, we can collect antibody from patients. So patients who have high titers of or high concentration of antibody specific for a particular antigen, let's say such as there's a very special infectious disease and we want to collect the serum of the patients and to find out that antibody as a treatment. So for example, if there's a hepatitis B as an antigen, then antibody against hepatitis B surface antigen, writing down HBsAg, and we can collect the serum of these patients and isolate those antibodies from this pooled plasma. And one of the risks is not only during this isolation, that carries the risk of other kinds of infectious disease can transmit. For example, like HIV, which will be a big problem. So this is one way to collect and gather antibodies. So another way is we can use animal. This is more typical. So such as mice or rabbit or goat. When you are purchasing antibody for experiment, you will find out these species. So how do we manufacture antibody? You know, antibodies are pretty expensive, and the reason is here because when we generate, we are intentionally exposing the animal to a specific antigen of interest or immunizing the animal, such as, let's say, hepatitis B surface antigen, we expose this one to those animals. And then we wait some time and we collect blood serum from that animals. And think about it, what kind of antibodies against these antigen will be collected? Then we have to consider about the shape of this antigen. Let's say these are the hepatitis B virus. Then this virus will have a different kind of a antigen or epitopes, different kind of epitopes. So these are as a foreign getting into the animal's immune system will develop antibody against these antigen, especially you can recognize there are multiple sites of epitope. So the blood serum from animal will contain not only single antibody against, but multiple. So that we call a polyclonal antibody. Because why do we say clonal is this antibody will be produced by many different B cell clones. Clone means let's say there is a very specific antigen against the antibody. The B cell will create clones of these the same antibodies, while another B cell will have another epitope will generate another clones of antibody. So this will cause differences in this polyclonal antibody will have differences in epitope, or because its epitopes are different, so each of these will have different affinity against the administered antigen. So what will be an issue from here compared to from patient? From patients, the antibodies are human antibody, while from animals, we can see the antibody, heavy and light chain, constant region will be from animal. and that it's in fact foreign to the patient for clinical use. So the caveat here is foreign antibodies can cause immune reactions. So it may be limiting treatment to a single exposure, because one single exposure will cause the human body create lots of antibody against this animal antibody, so that it may cause a immune reaction next time. So this is a diagram showing manufacturing antibodies, the effect. So x-axis is time as a week, y-axis is a logarithmic scale of antibody concentration in serum. So initial exposure will cause the B cell, specific B cell to produce antibody. So initial antibody on the surface of the B cell will create IgM. And then over time, IgG, the amount will be exponentially increased. And you can see that after injecting antigen A. And 6 weeks after, let's assume that we inject antigen A again. So what happens for this antibody against response to A is IgG is naturally decreased, and then this second exposure, it will boost up again. But you look at this slope is different. That is, the second time is much faster, because our body immune system will recognize this antigen A and be ready. The next exposure, it will be much faster to respond. It's because it's called the memory effect, because we remember what happened before. While if we happen to inject antigen A and B together at week of 6, the IgG or immunoglobulin G antibody against the B, the response will be not as stiff as this is the first exposure against antigen B. So the memory effect can be explained as specific antibodies are produced after exposure to antigens.